On July 21, 2024, Perseverance stopped moving. Not because it had failed, because its instruments had just registered something the science team could not immediately explain, without invoking a word NASA has spent 30 years refusing to say in public. The rover was parked in Neretva Vallis, an ancient river channel feeding into Jezero Crater. In front of it sat an arrowhead-shaped slab of mudstone named Cheyava Falls. Within hours, the team back on Earth had a problem. The chemistry did not fit the polite explanation. On our planet, the exact combination of features on that rock is produced by one process: life. Now, before we go further, tell me in the comments where you are watching from and whether you honestly believed we had found nothing on Mars yet. Because the record says otherwise, and it has been sitting in front of us since 2024. Here is what Cheyava Falls shows. Small dark rings, informally called leopard spots, distributed across the rock face. Fine veins of calcium sulfate cutting through the mudstone, meaning water flowed here for extended periods. Organic compounds detected by the Sherlock instrument, meaning carbon-based molecules of the kind biology uses. And in the tightest coincidence of all, iron phosphate and iron sulfide minerals sitting exactly where the leopard spots appear. Vivianite, greigite, two minerals that, in every terrestrial sediment ever studied, form as byproducts of microbial metabolism. Any one alone would be interesting. Together, in the same rock, in the same context, they form a pattern that geochemists on Earth would call a biosignature without hesitation. But this is not Earth. And NASA does not have the luxury of speaking plainly. The paper describing Cheyava Falls was published in Nature in September 2025. The language is careful, but it is not vague. The authors use the phrase potential biosignature. They rule out the easy abiotic explanations one by one. They admit the simplest reading of the data involves biology. Then they stop just short of saying it. The press release, meanwhile, calls the finding intriguing. It emphasizes the need for more study. All true, all also in a very specific way misleading. Because between the press release and the paper, there is a gap. And in that gap is the story no one at NASA is authorized to tell you directly. The public believes we are still searching. The data says we may have already found. The reason those two sentences do not match is not scientific. It is institutional. If Cheyava Falls is what the chemistry suggests it is, then the question is no longer whether life ever existed on Mars. The question is why NASA cannot afford to say so out loud. Read the NASA press release from September 2024 and then read the Nature paper from 2025 next to it. They describe the same rock. They do not tell the same story. The press release uses intriguing. It uses possible biosignature. It reminds you twice that only sample return can settle the question. Every sentence is technically correct. Every sentence is also engineered to prevent a headline. The paper is different. It walks through the abiotic explanations one by one. Hydrothermal alteration, late diagenetic fluids, high-temperature mineral formation. It shows that each requires conditions the surrounding sediment does not support. Then it states that the biotic explanation remains, quote, consistent with the observations. That is scientific language for the least uncomfortable interpretation involves life. Two documents, one event, two different registers. Why? Because NASA remembers 1996. That August, a team at Johnson Space Center held a press conference about a Martian meteorite designated ALH 84001. They said the rock contained possible fossilized microbes. President Clinton stood on the White House lawn and spoke to the nation about the discovery. Then it collapsed. Slowly, over years. Other labs replicated the mineral structures without biology. The organic compounds turned out to be explicable through non-biological pathways. By the early 2000s, the consensus had shifted from possible fossils to probably not. The institutional lesson was clear: never again. Not without samples in a laboratory on Earth. That wound shaped everything that followed. It shaped the 2019 methane detection, where Curiosity found seasonal spikes that could indicate biology, and NASA framed the announcement so cautiously, most viewers walked away thinking nothing had happened. It shaped the 2015 flowing water claim, later reinterpreted as dry granular flow. Each time, the pattern was the same. Show excitement, hedge the language. Let the public forget quietly when the finding softens. Cheyava Falls is different. The evidence is stronger, the context is better, the instruments are more precise. But the institutional muscle memory is the same. The problem is that public trust does not read nature papers. It reads press releases. And the press release is designed to protect NASA from another 1996, even if the cost of that protection is that the most significant discovery in the history of the agency gets described in the same tone as a mildly interesting geology update. To understand why this time actually is different, you have to understand what Cheyava Falls does that no rock before it has done. And that requires understanding the chemistry itself. Here is what is actually happening inside those leopard spots, and here is why, on Earth, no geologist would hesitate. A leopard spot is a chemical boundary. In the middle of the spot, the iron is in one oxidation state. Around the ring, it is in another. That transition from reduced to oxidized iron is not decorative. It is the fingerprint of an electron transfer. Something moved electrons from one atom to another across that boundary, and that something needed energy. On Earth, in sediments almost identical to what Perseverance is looking at, that energy comes from microbes. Specifically, from a class of bacteria that make their living by eating iron and phosphorus. They cannot use sunlight. They live in the dark, in the mud, at the bottom of lakes and swamps. They pull electrons out of iron minerals and dump them into phosphate or sulfate compounds nearby. The byproducts of that metabolism are precisely the two minerals Pixel detected in Cheyava Falls. Vivianite, a blue iron phosphate mineral. In terrestrial sediments, it forms in low oxygen environments where microbes are actively cycling phosphorus. Where you find vivianite, you almost always find fossilized microbial mats nearby. It is used as a diagnostic marker in paleobiology, precisely because its presence is so tightly coupled to biological activity. Greigite, an iron sulfide mineral. It is produced by sulfate-reducing bacteria, one of the oldest metabolic lineages on Earth. It shows up in wetland sediments, in the mud beneath anoxic ocean floors, in the mud beneath every bog on the planet. Some microbes even manufacture chains of greigite crystals inside their own cells to navigate along magnetic field lines. On Earth, greigite is not incidental to biology, it is downstream of it. Now think about what that means. Perseverance did not just detect one biosignature. It detected the byproducts of two different microbial metabolisms, sitting together, in the exact spatial pattern those metabolisms produce on Earth. Let that sit for a second. In every terrestrial sediment ever studied, that specific combination has one dominant author. There is a theoretical way to produce vivianite and greigite without life. It requires very specific pH conditions, elevated temperatures, and a source of dissolved sulfide and phosphate reacting with iron in a narrow chemical window. The pathway exists on paper. It has been produced, marginally, in laboratories, but it has never been observed to produce leopard spots. It has never been observed to produce them at the scale seen at Cheyava Falls. And it has never been observed in a sedimentary context resembling the one Perseverance is standing in. So there is an abiotic explanation. Technically, it exists. But it is a single explanation, threading a very narrow chemical needle against a biological explanation that fits every observation without strain. One story requires the universe to have done something rare and specific. The other requires Mars to have once been alive. Look at the one escape route the data leaves open, and then look at where the rock is sitting. The abiotic pathway for producing vivianite and greigite together requires three conditions in tight combination. First, elevated temperature, generally above 60 degrees Celsius and often much higher. Second, acidic or strongly reducing water chemistry with a specific sulfide activity range. Third, sustained fluid flow through the rock long enough for the crystals to nucleate and grow. Meet those three conditions, and you can produce these minerals in a lab without any biology involved. Now look at Jezero Crater. Jezero was a lake, not a hot spring, not a hydrothermal vent field, a lake, fed by a river system flowing off the crater rim into a delta that spread across the basin floor. The sediments Perseverance is driving through are the ancient bottom of that lake. They preserve fine mudstone layers, evidence of slow settling in calm water. They preserve carbonate minerals, which form in neutral to mildly alkaline conditions. They preserve clays that only stabilize at moderate temperatures. None of those signatures are compatible with a hot, acidic, hydrothermal environment. The rock is telling us the water at Cheyava Falls was cool. It was near neutral in pH. It was quiet. That is a problem for the abiotic explanation, because the abiotic pathway needs exactly the conditions the surrounding sediment says were not present. You cannot have it both ways. Either the mudstone is lying about the depositional environment, or the abiotic explanation for the leopard spots is lying about how the minerals formed. And mudstones do not lie easily. Their chemistry is stable, well understood, and cross-checked across dozens of terrestrial analogues. This is the same logic Earth geochemists use to confirm biosignatures in ancient rocks. In the Pilbara region of Western Australia, 3.5 billion-year-old sediments contain structures that resemble microbial mats. For decades, researchers debated whether the structures were biological or the result of exotic mineral processes. What settled it was context. The surrounding sediments showed the environment was too calm, too neutral, too shallow for the abiotic pathways to work. Biology became the simplest remaining explanation. Chayava Falls is being read with the same framework, and the framework is producing the same answer. The abiotic explanation is not impossible. Nothing in science is impossible, but it requires the local environment to have been dramatically different from what every other line of evidence in Jezero shows. It requires a specific chemical event to have happened in a specific place, leaving no trace anywhere else in the crater, and producing a pattern that coincidentally mimics the fingerprint of biology. That is not a competing hypothesis. That is a coincidence stacked on a coincidence, defended by silence. And now the problem stops being about one rock. One rock is a curiosity. A pattern is a verdict. Cheyava Falls is not sitting alone. It is embedded in a geological formation called Bright Angel, a light-toned band of sedimentary rock that stretches for kilometers along the Neretva Vallis Channel. The same instruments that flagged Cheyava Falls have been sampling other outcrops within Bright Angel throughout 2024 and 2025. And the pattern is not going away. Multiple rocks, same formation, similar mineral signatures, similar leopard spot morphology, similar iron phosphate and iron sulfide correlations. The team has been careful in public statements, but the paper is direct. What Perseverance found at Cheyava Falls is not an isolated feature. It is representative of a wider chemistry across the entire Bright Angel Formation. Now think about what that means. A single anomaly is easy to explain away as a statistical accident. Even the strictest geochemist admits that unusual mineral combinations can occur once in one rock by chance. But when the same combination shows up across kilometers of the same sedimentary layer, chance stops being a serious explanation. You are no longer looking at a curiosity, you are looking at a signal. And then it gets worse. Roughly 3,700 kilometers away, in Jezero Crater, the Curiosity rover has spent over a decade studying a completely different ancient lake environment. Curiosity has reported organic compounds, seasonal methane variations, and mudstone chemistry that repeatedly hints at the same kind of low-oxygen, carbon cycling conditions that dominate microbial habitats on Earth. The two craters were formed by different impacts, filled by different water sources, and studied by different missions. But the underlying story keeps rhyming. Two craters, two river systems, two ancient lakes, two independent lines of evidence pointing in the same direction. The implication is not subtle. If Cheyava Falls represents biology, it was not one lucky puddle where life briefly appeared. It was part of a widespread environment. Whatever produced those minerals was doing so across large areas over long periods in multiple basins. Which means Mars was not almost habitable once. It was habitable, and something was living in it. Let that image form in your mind. Not a dead planet with one weird rock. A planet with lakes and rivers and mudflats and shallow deltas, all of them wet, all of them chemically alive with microbial cycling for hundreds of millions of years. A world with a biosphere. Small, simple, probably confined to sediments and shorelines, but real. And then, at some point, it stopped. That is the frame the evidence is pushing us toward. Not a puzzle about one rock, a cemetery the size of a planet. If Mars was a cemetery, then the next question is not academic. It is diagnostic. It is the question every biologist and every planetary scientist should be asking right now. How did it die? A planet does not die in one moment. It suffocates. Around 4 billion years ago, Mars had something Earth still has, and it was the difference between a living world and a dead one. Mars had a global magnetic field. Deep in its core, molten iron was moving, generating an invisible shield that wrapped around the entire planet. That shield deflected the solar wind, the constant stream of charged particles pouring out of the sun. Without that shield, the solar wind would strip the atmosphere. With it, the atmosphere stayed put. Then the core cooled. The dynamo stopped. The shield vanished. We do not know exactly why. Mars is smaller than Earth, so it lost internal heat faster. It may have had a different mantle composition. The details are still debated. What is not debated is what happened next. The solar wind hit the upper atmosphere directly. Charged particles peeled off molecules of nitrogen, oxygen, carbon dioxide, and water vapor. Slowly at first, then relentlessly, over hundreds of millions of years, the atmosphere bled into space. The MAVEN spacecraft has been measuring this process in real time since 2014. Mars is still losing atmosphere today. Roughly 100 grams per second, escaping into the void. 4 billion years after the dynamo failed. That is a small number in the moment. Multiply it across geological time, and you get an entire ocean of gas. Gone. As the atmosphere thinned, the pressure dropped. Liquid water became unstable at the surface. It either froze into the subsurface or sublimated directly into vapor, then escaped along with everything else. The rivers stopped flowing, the lakes evaporated. The mudflats where microbes may have lived dried and cracked and buried themselves under dust. If life existed on Mars, this is how it ended. Not in fire, not in a single catastrophe, in a long, slow strangulation, as the physical conditions that made life possible were peeled away one molecule at a time. Now hold that image, because here is what should bother you. Earth has the same kind of shield. Our magnetic field, generated by the same kind of molten iron dynamo, is the only thing standing between us and the same process. And that field is not permanent. Over the past 200 years, it has weakened by roughly 9%. The magnetic North Pole has been drifting across the Arctic at accelerating speeds. Weak spots have opened over the South Atlantic, where satellites already have to power down when they fly through. None of this means Earth is about to lose its atmosphere. The timescales are long. The system is more robust than Mars ever was. But Mars is showing us, in real time, what happens when the protection eventually fails. And that lesson is not about the past. It is about what a planet like ours looks like at the end. Perseverance has been doing something almost no one talks about. Since 2021, it has been drilling core samples, sealing them in titanium tubes about the size of a piece of chalk, and dropping them at cache sites on the Martian surface. As of late 2025, more than 30 of those tubes are sitting there. One of them, Sapphire Canyon, contains material drilled directly from Cheyava Falls. That tube may be the most valuable physical object in the solar system that is not on Earth. And right now, it is stuck. The plan to bring the samples home is called Mars Sample Return. When it was announced, the projected cost was around $7 billion. The architecture involved a NASA lander, a European return orbiter, a Mars ascent vehicle to launch the samples off the surface, and a rendezvous in Mars orbit, followed by a return to Earth. By 2023, cost projections had climbed past $11 billion. Timelines slipped from a 2033 return to sometime in the 2040s. In April 2024, NASA effectively paused the existing architecture and asked industry to propose alternatives. The message was clear. The original plan was unaffordable, and nobody had a cheaper one ready. The samples are still in Jezero. Perseverance is still driving. The tubes are still sealed. And the mission that was supposed to bring them home is in indefinite limbo. Consider what that actually means. The single most important scientific question of the century, whether life has ever existed beyond Earth, may be answerable. The evidence may already be collected. It may be sitting in a titanium tube at a spot called Three Forks, waiting for a spacecraft that may not launch this decade or the next. The scientific bottleneck is not the science. What is missing is the rocket. And here is the harder part. Even if a sample return mission launches in the 2030s, the return would not happen until the late 2030s at the earliest. Analysis in Earth laboratories would take years. Confirmation of biosignatures, if confirmation is possible, might not arrive until the 2040s. Some of the scientists who found Cheyava Falls will be retired. Some will not be alive. An answer that could reshape human self-understanding is being throttled by budget cycles and launch cadence. That is not a scientific problem. It is a civilizational one. And while the samples wait, the public message keeps softening. Because if you cannot confirm, you cannot claim. Which brings us to the last layer of the story. In 2021, NASA published a framework called the Confidence of Life Detection Scale, or COLD. It runs from Level 1 to Level 7. Level 1 is a preliminary signal. Level 7 is confirmed detection of extraterrestrial life. Every level in between represents a specific standard of evidence that has to be met before a claim can be advanced. The scale exists for one reason. NASA needed a way to talk about biosignatures without ever again saying more than "the evidence supports." Where does Cheyava Falls sit on that scale? Public statements suggest Level 1, a preliminary signal. Interesting, worth studying, not yet meaningful. That is the register used in press releases, in NASA briefings, in official quotes. The paper, read carefully, describes something closer to Level 2 or Level 3. Level 2 requires that the signal be reproduced and characterized in detail. Level 3 requires that abiotic explanations be systematically evaluated and, where possible, ruled out. Both of those thresholds have been substantively met at Cheyava Falls. The paper documents the signal in multiple instruments. It walks through the abiotic explanations. It shows why they do not fit the environment. So why does the public register lag behind the technical one? Because Level 4 requires confirmation from an independent instrument, and Level 5 requires confirmation from an independent sample. And neither is currently possible without Mars sample return. The moment NASA advances the claim above the level it can defend, it exposes itself to another 1996. That is the memory that shapes the language, not just ALH 84001, the 2015 recurring slope lineae, announced as evidence of flowing water and later reinterpreted as dry granular flow. The 2019 methane spikes at Gale, announced as potentially biological and still, 5 years later, unexplained but unclaimed. Each retraction cost credibility. Each cost budget leverage in Congress. Each made the next announcement more cautious than the last. To be fair to NASA, this is not a conspiracy. It is not scientists hiding results. The paper was published in Nature. The data is public. Nothing is being suppressed. What is happening is quieter and more human. Institutions respond to incentives, and the incentives at a publicly funded agency that has to justify multi-billion-dollar missions to skeptical politicians favor caution over precision. The public does not receive a false version of reality. It receives an edited one. Edited not by malice, but by institutional fear. And behind that fear is something bigger than the fear itself. Something that reshapes what finding life beyond Earth actually means. Because if Mars had life, we are no longer alone in the way we thought we were. Here is the part that reorders the story. For most of human history, we assumed that life was special, that the origin of biology was a rare event, maybe unique to Earth, that the universe was mostly empty because making a living cell was mostly impossible. If Cheyava Falls represents biology, that assumption breaks, because Mars and Earth are neighbors. They formed from the same solar disk at roughly the same time from similar raw materials. They both had liquid water on their surfaces during the first billion years. If life arose independently on both, in the same solar system, then the transition from chemistry to biology is not rare. It is what wet, rocky planets do. And if it is what wet, rocky planets do, the galaxy is not empty. It is full. That reframes an old problem. In 1950, physicist Enrico Fermi asked a question that still has no accepted answer. If the galaxy is full of stars, and many of those stars have planets, and biology is not that hard, where is everyone? Why do we see no signals, no probes, no evidence of any technological civilization? The question has many possible answers. Maybe intelligent life is rare, even when microbial life is common. Maybe civilizations do not last long enough to be detected. Maybe interstellar travel is harder than we think, and everyone stays home. Any of these could be true, and honest scientists disagree about which is most likely. But one framing that has gained traction is the idea of a great filter. Somewhere along the path from simple chemistry to spacefaring civilization, there is a barrier. Something that stops most life from getting past a certain stage. The question is where that barrier is. Behind us, meaning we already passed it and are unusually lucky. Or ahead of us, meaning we have not hit it yet. If Mars had life and Mars died anyway, that shifts the weight. It suggests the origin of life is not the filter. Simple biology is common, something else stops it. And that something else might not be behind us, it might be waiting. This is not certainty. It is one interpretive frame among several. Reasonable people read the same evidence and reach different conclusions about what it means for our own future. But it is a frame that becomes harder to dismiss the more evidence for extraterrestrial biology accumulates. Copernicus taught us Earth was not the center of the universe. Darwin taught us we were not separate from other animals. Each of those shifts felt uncomfortable at the time. Each of them turned out to be true, and each of them made the next question harder. Perseverance may be about to teach us something similar, that we are not the only ones this planet has been, and that is the part nobody wants to say out loud. So let us come back to the rock. On July 21st, 2024, Perseverance stopped moving. It stopped because its instruments had just registered a combination of features that, on our planet, has only one known author. Iron in two oxidation states across a chemical boundary. Vivianite and greigite in the exact spatial pattern that microbial metabolism produces. Organic compounds nearby. Calcium sulfate veins showing that water was present long enough to matter. A mudstone context that rules out the hot, acidic conditions the abiotic pathway would require. And a pattern that does not appear in one rock, but across kilometers of the Bright Angel Formation, with echoes in Jezero Crater 3,700 kilometers away. 3 pillars. Chemistry that biology writes almost exclusively. An environment that closes off the non-biological escape route. A geographic distribution too wide to be coincidence. That is the case. Read honestly, it points in one direction. The most honest interpretation of the evidence, not the safest one, is that Mars once had life. Simple, microbial, sediment-dwelling life. It lived in the mud at the bottom of lakes. It cycled iron and sulfur and phosphorus. It left the same chemical fingerprints its terrestrial cousins leave in the mud of every wetland on Earth. And then, over hundreds of millions of years, as the atmosphere bled into space, it died with the planet. That interpretation may be wrong. Science works by trying to prove itself wrong. Confirmation requires samples in a laboratory on Earth, and those samples are stranded, waiting for a mission that may not launch in this decade. But the reassuring frame that we have not found anything yet, that the search continues, that this is just another intriguing hint, is not honest anymore. We have found something. We are choosing not to call it by its name. And the reasons for that choice are institutional, not scientific. The science is done. The rocks have spoken. What is left is the courage to say what they said. If we ever bring those tubes home, and if the terrestrial analysis confirms what the Martian data already suggests, then the year 2024 will be remembered as the moment a rover stopped in front of a mudstone slab and quietly registered the most important finding in the history of biology. It just took the rest of us a while to catch up. Until then, the rock sits under the Martian sun, silent, sealed, waiting. NASA's caution does not change what is written in that rock. It only delays the moment when we will have to accept it.